Hi, today I am going to present one of the most interesting field or demand of bioinformatician or the biologists or who are interested in computational biology or bioinformatics. And that is branches of bioinformatics and introduction to bioinformatics. This is very important for the perspective of higher education and for the perspective of the jobs in the companies or industries. So this is very interesting topic, and I could say it is highly demanding. Here, basically, I'm going to introduce the basics of bioinformatics, which is an interdisciplinary field that combines biology, computer science, statistics, and information technology to analyze and interpret large biological data sets. Key branches include genomics, transcriptomics, and the functional genomics. Other areas include structural biogenomics, comparative genomics, and systems biology. It develops and uses computational tools and methods to analyze, interpret, and manage biological data. It combines the principles from biology, computer science, mathematics, and statistics to make the sense of the vast amount of information generated by modern molecular biology techniques, such as genome sequencing. The core functions of bioinformatics include creating and maintaining biological databases to store and organize biological information, developing algorithms and software for the analysis of large-scale data sets, interpreting the results to gain new biological insights. And you can see here the application, what it requires for especially for bioinformatics. You need to have the knowledge of computer science, chemistry, biochemistry, biology, statistics, mathematics, and slight basics of engineering. Especially, I also want to clarify that the people become fearful about knowing that so many fields you are required. But please be aware that only basics are required of all these seven fields. It is not needed for highest level information, even intermediate level or plus two level information or knowledge of these subjects are more than sufficient to successfully work in this field or successfully continue the higher education in bioinformatics. Now, the key branches of bioinformatics. Bioinformatics encompasses several specialized branches, often referred to as omics, that each focus on a different layer of biological information. In genomics, the study of an organism's complete set of genes, including their structure, function, and evolution and mapping. Proteomics, the large-scale study of proteins, including their structure, functions, and interactions within a biological systems. Transcriptomics focuses on the complete set of RNA molecules produced by the cells of an organism, providing insights into gene expression. Functional genomics aims to identify the functions of the genes and their corresponding proteins, studying their roles and the interactions. Structural genomics involves determination of the three-dimensional structures of the proteins, which is crucial for understanding their functions. Comparative genomics compares the genomes of different species or organisms to understand evolutionary relationship and the genetic basis of traits. Systems biology uses mathematical modeling to analyze large sets of biological data and understand the complex interactions within a biological system. Chemoinformatics organizes and analyzes chemical data, often focuses on natural products with therapeutic potentials, but of course, synthetic drugs are also common, so it is not limited to natural products or natural drugs only. This is the summarized view in the picture where you can think about the diverse application of bioinformatics, for example, molecular biology, genomics, proteomics, and the multiomics things, computer-aided drug design, application or databases, computational resource development also, information technology and management also. Now, the key branches could be summarized. in different way depending on the job's requirements and the field where you are applying. For example, sequence analysis, gene expression analysis, medical bioinformatics, computational biology, biostatisticians, genomics, proteomics, transcriptomics, metabolomics, epigenomics, system biology. Now, biostatistics, machine learning in bioinformatics, biological data mining, phylogenetics, and structural bioinformatics. These are summarized branches of bioinformatics where you can get the large scale jobs in academia as well as the industries. Now, first application, that is genomics, which is highly common and frequent in research as well as the industry. Genomics is the study of the entire genome of an organism, including its genes, their interactions, and their influence of genetic variations on traits and diseases. Gene identification and annotations are included, locating genes and other functional regions within a genome. Comparative genomics, analyzing and comparing the genomes of different species to understand the evolutionary relationships and biological differences. Pangenomics, studying the complete gene set of a species by analyzing all the genetic materials across a given taxonomic group. So here in the figure, I have summarized the different applications in genomics era: whole genome sequencing, genome biology, host pathogen genomics, transcriptomics, host transcriptional responses, gene expression signatures, ex evolutionary histories, phylogenetic divergence, transmission dynamics, outbreaks, genome-wide association studies, candidate gene studies, genetic diversity, genomic variations, pathogen molecular physiology, detecting of drug resistance mutations, drug resistance surveillance, treatment guidelines, drug discovery, and vaccine development. Now, in proteomics, you can think about the large-scale protein or peptide identification. So proteomics is the large-scale study of entire set of proteins or the proteomes expressed by an organism or by a cell of an organism. Proteins are the primary functional molecules in the cells, and proteomics, it aims to understand their structure, function, and interactions. So in protein identification and quantifications, using techniques like mass spectrometry to identify and measure the level of different proteins in a sample, which is quite time taken process, but it is commonly used. The structural proteomics, predicting and modeling the three-dimensional structures of the proteins, analysis of the protein interactions, studying how proteins interact with each other to form cellular networks. So these are the summarized application which you can see here. For example, Geoterm, PFAM, KEG, Reactome, HPRD, Interpro, String, these are the databases which you are using for protein functional assay, cell tissue functional analysis, protein assemblies, proteins, peptides, LCMS, MS, and many more kind of identification. Now, you can think about the types of proteomics workflow depending on the exam machines which you are going to use for protein identification. Are you going to work for limited number of peptide or protein identification, or you are going to hold proteomics identification and characterizations? This depends on you, and based on that you can prepare the workflow for proteins identification. Now, transcriptomics, which is very common and frequently used, especially in academia for research purpose. Transcriptomics is the study of full set of RNA transcripts or the transcriptome in a cell under specific conditions. It reveals which genes are active and how their expression levels changes in different conditions. Gene expression analysis using techniques like RNA sequencing and microarray analysis compare gene expression patterns between different cell types or disease states. Regulatory analysis investigating the mechanisms that control the gene transcription. And in the right side, you can see the advanced level application in transcriptomics for the bioinformatics tools and techniques or your own developed methods. For example, here, molecular immunophenotypes identification. This is one way to identify. Figure B is co-expression networks of the genes, and then upstream mechanisms, then single cell transcriptomics, and finally personalized transcriptomics for clinical diagnostic purpose. Now, you can think about also metabolomics, which is also commonly used, especially for those people who are working in plant or environmental science. Metabolomics is the study of a small molecule metabolites found within a biological system, such as a cell, tissue, or organism. Metabolites are the end products of cellular processes. Here, pathway analysis, analyzing metabolic networks to the understand biochemical processes, diagnostic biomarkers, identifying the metabolites associated with diseases for diagnostic purposes. If you want to see all the application in terms of research paper, I have my own publication for almost all the system biology and bioinformatics branches application, which you can see on my Google profile, Google Scholar profile, or you can see on the ResearchGate or some other profiles, and you can just search my complete name, Mohammad Mobashir, and you can find all the research papers and diverse application of bioinformatics for research paper publications. Now you can think about the structural bioinformatics branch. This branch is focused on the computational modeling and analysis of three-dimensional structures of biological macromolecules such as the proteins and nucleic acids, protein structure prediction, predicting the 3D structure of a protein from its amino acid sequence, and then molecular docking, simulating how small molecules such as drugs may bind to the target proteins. So there are a number of softwares or web servers available for molecular docking as well as three-dimensional structure prediction. But for three-dimensional structure prediction these days, AlphaFold 3, which is the advanced and one of the best in terms of accuracy software with respect to almost all the existing softwares. So you can predict the 3D structure of any kind of proteins by using AlphaFold 3, and you can get one of the amazing result with respect to accuracy. Of course, you can use the other previously or existing softwares for the same purpose. Just you need to choose if you are going to work on homology modeling or de novo prediction. It's your choice, or you can also develop your own software. So if you are a good bioinformatician, then you can also try to find the new way of prediction of the software of the three-dimensional structure, and you can use the advanced version of molecular machine learning tools or AI applications for predicting the 3D structures of the protein. Now, clinical bioinformatics applies the computational methods to analyze clinical and genomic data to support disease diagnosis, treatment, and prevention. Personalized medicine is one of the major goal for the scientists to apply bioinformatics using an individual's genetic profile to tailor medical treatments, cancer genomics identification of mutations, or the other changes at epigenomics level in tumor genomes to guide the targeted therapies, infectious disease tracking, using pathogen genome sequences to track outbreaks and the spread of antibiotic resistance. You can perform the infectious disease tracking, and this is the map here figure is clearly showing the application of bioinformatics for clinical genomics, personalized medicine, pharmacogenomics, and genetic epidemiology. So this is called translational bioinformatics, or you can also say the health bioinformatics. It depends on you. If you want to summarize the clinical bioinformatics application, then the right side figure here, you can say data analysis, interpretation, precision medication, and improved diagnosis, and then clinical implications, translational research, prediction, drug design, bioinformatics, clinical informatics. You need to have good sets of tools, databases, and prior knowledge of the basis also. So this is why what you need for clinical bioinformatics, and this is quite easy to work in clinical bioinformatics sector. Now, computationally evolutionary computational evolutionary biology. That means you are going to establish the relationship between different organisms and the going to predict the minute level differences between one organism to another organism. Or you can also work on how the cancer cells have evolved from normal cells. So this field uses computational techniques to study the evolutionary relationship between the organisms by measuring changes in their DNA over time. Phylogenetic analysis is one of the first steps in case of evolutionary systems biology, where you are constructing the evolutionary trees to trace the origin and descent of a species. Comparative genomics, using genome comparisons to trace complex evolutionary events like gene duplication. And when the people are talking, in addition, third uh, subtopic of computational evolutionary biology is basically when you are going to predict the driver and passenger genes in case of cancer or any disease, you are going to apply the similar approach. For example, if you are working on cancer, you need to predict the driver and passenger genes, then you need to construct the phylogenetic trees and find those genes which are mutated in case of or because of cancer. Then you can say that these are something which is called driver genes or driver mutations in case of different types of cancer. But if you want to see the detailed application, you need to see the research papers. There are a number of research papers available for driver and passenger gene prediction, and this is also very similar and easy part of computational evolutionary biology or bioinformatics applied in case of prediction of evolutionary relationship. Now, other notable branches is basically which I have mentioned. It's chemoinformatics, which is very common and frequent in case of structural bioinformatics or structural biology, and biodiversity informatics, which you can see here the how and where you are going to apply the bioinformatics for different purposes. So you can study the soil, you can study the diverse forests, you can study the river, you can study the soil samples, you can study the sea samples, you can study the sea waters for contamination purpose or identify new organisms. Then it comes to systems biology, which is here. You can see where you are using computational and mathematical modeling to analyze and simulate the complex interactions within the entire biological systems, where you have some kind of input information, some kind of sensor development, some kind of signaling networks, gene regulatory network, metabolic networks, and transcriptional networks. Then you focus on the phenotypes or output response. So it depends on you and your choice. So where you are going to work and how you are going to work. Now, then it comes to integrated bioinformatics, which is basically the part of system biology where you are going to read the scientific literature, then go for analytical in silico techniques, molecular data, clinical data, biological modeling, pharmacological data, and then finally look for some kind of predictions for the desired project goals. So if you want to understand the evolution in case of bioinformatics from where we got to know or study the new subject field, that is bioinformatics. So it started from 1990s, and in 2001 it was full-fledged bioinformatics course, and this is the complete overview of how bioinformatics or computational systems biology has evolved and how it could play or it is playing the potential roles in the advancement of biology, in the advancement of diagnostic sectors, in the advancement of therapeutic purpose. So, what you need to know in addition in bioinformatics, the most important thing is to understand and know the major biological databases. For example, NCBI is one of the biggest database for biology and oldest database. And here then it comes to some kind of bibliographic database, sequence database, structure database, metabolic database, then chemical database, disease database, taxonomical database, microarray database, enzyme there are hundreds of databases which you need to be familiar depending on your requirement and its project requirements and the complete designed goal of your project. So a database is an organized collection of related biological information that can be easily stored, accessed, and managed. So this is what plays crucial role if you are familiar with the relevant databases which you can access from time to time. Now, if we are going to summarize the top emerging trends or fields or branches of bioinformatics, then we can say single cell omics, multi-omics, QSAR, structural bioinformatics, and protein and drug design, spatial transcriptomics and imaging, database development, quantum computing in bioinformatics, blockchain for securing the data sharing, AI, artificial intelligence, and machine learning approaches in bioinformatics. Finally, I would like to say thank you for patience and watching our channel and these videos. And please don't forget to subscribe our channels, which are here, Change by Science and Bioinformatics for All.